Hello, everybody. It is my pleasure today to introduce to you Dr. Silvia De Monte, who has graciously accepted the invitation from the Physical Biological Interaction Group within the framework of the Scholar Program, the María de Maeztu. Silvia is a leading researcher in the field of collective behavior of biological populations. She is a senior researcher at the Institute of Biologie de l'École Normale Supérieure.
Very good.
in Paris, actually, where she's member of the Evo Math group, and she also collaborates with the Max Planck Institute for Evolutionary Biology in Plön in Germany. She has a background in physics and biology. She earned her PhD in physics from the Technical University of Denmark, where she investigated the dynamics of globally coupled systems and the emergence of collective behavior in heterogeneous populations. Her research focuses on the evolution of collective behavior in microbial communities, on evolutionary dynamics and nested Darwinian populations, on the mathematical modeling of cooperation, of cheating, of self-organization, and also on plankton biogeography and species abundance distributions in marine ecosystems. She has published extensively in high-impact journals such as Nature, Ecology and Evolution, eLife, PLOS Computational Biology, PNAL, and her work that bridges theoretical biology and experimental data, bring deep insight into how selection operates at multiple levels of biological organization. And with that, I will welcome Silvia and thank her for being here today.
Thanks a lot. Wow. Thank you very much for this nicest introduction. Indeed, in my research, so my passion is really to understand how microbial populations manage to work together despite diversity and heterogeneity. But today I will focus on one specific project where we have been, that is a part of a broader research interest on how microbial communities work at the level of a community. So as you probably all know, microbial communities are essential for many functions that are not provided by just one species in the community, like, for instance, nitrogen fixation in the soil, the equilibrium of the global cycles for phytoplankton, or even human health for host-associated microbiomes. And in this world of microbes that are very diverse has been sort of very partially known until very recent years when people have started to use genetic methods, genomic methods to characterize the diversity of microbial ecosystems. In this moment, since sequencing has become very common and available broadly, there's a lot of exploration of this diversity, but we still don't know many things like the response of diversity of microbial communities to climate change rather than to antibiotics treatments and all this. What is also very interesting of microbial communities is that they can vary variously. I mean, they can be maintained in the lab. They can be maintained in the lab also in case of assemblages of species, so not single species cultures, but the coexistence of many different species. And therefore, they can be, we start to be in a place where we can ask quantitative questions about their behavior, the processes that underpin the dynamics, the ecological and evolutionary dynamics in the lab and in the wild. The main problem associated with this is that with an estimated 10 to the 29 cells of Earth, of which belong to millions of different species, and you can imagine how many different kind of interactions occur among all these organisms, we don't really know where to start with describing these systems if we want to encompass all the diversity that they actually possess. So the kind of questions that I'll be asking today, and that many people are asking around the world, is what is there of general that can be said about this kind of ecosystems, and potentially whether one could use these general features to infer what are the common processes that shape ecosystems, and possibly also how they change when you change, I don't know, environmental conditions, for instance. Three really key features that are observed, I would say universally, let's say ubiquitously in microbial communities are their diversity, what is called the commonness of rarity, so there are many, many rare species, and the existence of a very strong turnover of different species. Let me be a bit more specific about this. So you have probably heard about the rare biosphere or microbial dark matter. So this is the ensemble of species that are present in an ecosystem, but with very, very small abundance. So if you rank species, let's say you have an ecosystem and you measure how many individuals are there for every species. For microbes, the definition of species can be different depending on how you deal with the data, but let's say you choose a method and you count how many individuals belong to that cluster, that type, that you can call a species. And then you can rank the species from the most abundant to the least abundant. What happens is, of course, this is a decreasing function, but if you look macroorganisms, what will happen in general is that you have relatively low diversity communities. For instance, if you rank, I don't know, bird species, or also trees in temperate latitudes, you would find that actually you have a relatively small number of, I mean, you always have many more rare species than abundant ones, but most of the community is made of abundant species. On the contrary, microbial systems are characterized by the existence of a heavy tail of rare species that are very numerous and where people are not exactly sure what they're doing there. So are they going to die soon? Did they just emerge? Or are they essential or not? So there is a lot of debate in this moment about what is the role of these rare species. But this feature is really very, very common, and it is what underpins the high diversity of microbial ecosystems. The other observation that I mentioned before, and this is shared by any kind of ecosystem, is that in general you have many more rare species than you have common ones. And finally, this feature of the existence of many more rare species than common ones has been related to some temporal patterns. So typically, these rare species seem to be species that hang around for a long time and then every now and then become very common. And actually, most of the species have this kind of behavior. So for this reason, the rare biosphere has also been, so one of the most, let's say, most followed ideas is that the rare biosphere corresponds to a seed bank. So many species that are there, for instance, in planktonic populations, are actually not actively growing, they're just hanging around, and when the moments are favorable, then they will bloom and then become common. So all these different features are typically studied by looking at the distribution of abundances. So as I was telling before, in general, if you have a sample or a field patch, what you can do is to count how many individuals belong to every species. And then this is basically the most accurate kind of information that you can have if what you are looking at is just a species. And all the indexes like the Shannon index, species richness, whatever you want, evenness coefficients are just derived, they are statistics over the distribution of abundances. For this reason, people have been studying a lot these abundance distributions in an attempt to infer what were the processes that govern the distribution of these abundances. And just to give you an example, so this has led to a never-ending debate about which are the best models that describe a given abundance distribution. So this is a simulated distribution, but actually it looks very much like the Barro Colorado Island data set. That is a very famous data set where people have censused all trees in an island, all trees that have a diameter more than 10 centimeters. So every year people go there and count every single tree. So they have now for many years these distributions of abundances. And then what you typically observe is that you, for macroorganisms at least, is that you have some kind of peak at intermediate abundances. So here the most frequent class of abundance is not the rare ones, but those with intermediate abundance. So let me explain just how this is plotted. So instead of ranking, as I showed you before, species from the most abundant to the least abundant, what you're doing here is that you put abundance on the x-axis, and then you define a bin. For instance, this is individuals that have abundance 1 or 2. These are those that have abundance 3 or 4. And this is up to 8, and so on. So you have a bin of abundances, and then you count how many species are within that bin of abundance. Why do people use this representation and not the other one? Is mostly because this smoothens out a bit the data that are, for macroorganisms, pretty noisy. And also because you can compare these abundances directly to models. And then people have been discussing, actually, this one is better described by, I don't know, this certainly not this curve, but then this or this. So this can go on forever, and it has gone on forever. So there are out there many different models that produce distributions that look more or less like this. And then there are many statistical methods that you can use to fit this data. And then people, based on this, they conclude that, I don't know, some processes like competition between species are important or not. And now just to give you a concrete example, these are data from a data set that we have analyzed that comes from the Tara Ocean expedition, which you probably know of here. So this was an expedition that sampled the global ocean, and they provided abundances of operational taxonomic units that were sampled and processed in exactly the same way. So different samples were comparable. And now this is how a species abundance distribution looks like for a typical sample. So you would have a part of it that is pretty regular across samples, and the most abundant species, so these are the rare species, and these are the most abundant species, is usually very variable from one sample to the other. So what we wanted to do was to compare quantitatively the slope of this distribution. First of all, determine whether this was, really a power law distribution as it looks like in this example. And then we thought, okay, if this is a power law distribution, let's use the exponent of this distribution as a way to decide whether an ecosystem is species diverse or not. So in general, if this decays, if the decay is lower, then you would expect that the community is less diverse than if it is faster. So we did this with this idea in mind of using this as a proxy that was more reliable than the Shannon index. And it turns out that if you do this fit properly, you end up with an exponent that has a tiny variability. So it's less than 10% coefficient of variation across all the 390 samples that we had that came from 120-something different stations in the ocean. So somehow the rare biosphere is distributed in a similar way everywhere, whereas this part of very abundant species is what maintains, what keeps the information of biogeography. So this one changes from place to place. And all the rest seems to be basically invariant. So this tail of rare species is actually like 99% of the species. So it's not like a small fraction. So this was basically spurred us to ask a number of different questions. Before I go here, but what we were not very satisfied about is that in order to fit this distribution, we had to come up with some functional form. And what we chose is this negative beta binomial distribution that comes from a neutral or a semi-neutral model. So a model where every species is supposed to be equivalent to every other species. And with this distribution, the good part of it is that when the abundance is large, it is basically the product of an exponential and a power law, so you can simultaneously fit different kinds of distributions. So this distribution has been then renamed or called by Gao and others. Recently they called it power band. And they applied this power band to, several tens of thousands different samples that they had from animal, plants, microbes. And what they concluded is that it is the best distribution that shapes all these SADs. So this is the new baby of, this should be the best explanation for everything but there's other studies that actually find something different. So traditionally, you would not find this one. Traditionally, for a macroorganism, you would fit things with a unimodal distribution. And a unimodal distribution is found actually also in microbes by other studies that have looked at, especially at how the abundance of species vary across samples that are supposed to be samples of the same community. In this case, I don't know, for instance, gut microbes, if people assume that every gut is alike, then you can sample different people and look at the distribution of, for instance, of one single species across people and compare this with the distribution of many different species in one single person, for instance. And in this kind of studies, they seem to find the usual, unimodal distribution. So this was a bit bothering because it feels like everybody is fitting everything. So here you see that they're using basically the same data. And how is it possible that some people find one shape and some other people find a completely different shape? So this is what led us to trying to step back and think what were actually the things that every model had in common, and whether we could link these features to the shape that came out then from the model. So this work that I will present has been mostly thought of and realized by Emil Malmin, who is a super brilliant postdoc, who I have the pleasure to co-direct with Arne Tausen, the director of my department of the Max Planck. So to make a long story short, we realized that many models had as a central feature the fact that the birth rate, or if you want, the fitness of different species was fluctuating. This is known, for many reasons. It can come from environmental fluctuations. It can come from like statistical fluctuations when populations are small. It can come from the interaction with other species or with temporal fluctuations of interactions. So there are many ways you get fluctuating environments which reflect, so biotic or abiotic environment, which reflect into fluctuations in the growth rate of species. So what Emil did was that he started, look, oh, what did I do? Okay, we're back. I pressed the self-destruction button. So he started from a very simple equation. He said, okay, let's describe the abundance of every species, the variation in abundance of every species as a per capita rate. So you see that you have this proportionality to the abundance itself, and this is the rate. And this rate is made up of three parts. The first part is an instantaneous growth rate or instantaneous fitness, if you want. And this one can be described as a, he described it with a fluctuating, value that is ruled by, it's a color noise basically. It's an Ornstein-Ulbeck, I can't pronounce that name, process. So it's a time correlated noise. And on top of this, he said, okay, we need to avoid that a community grows indefinitely. So typically you would have interactions with many different species, but here he only introduced, some kind of community level limitation. So when the sum, big N is the sum of all Ni's, when the sum of all abundances exceeds a certain value, a certain carrying capacity, then this growth rate will become negative. And then you can add other features like self-limitation. This is typically a typical hypothesis in ecological models that an individual competes more strongly with individuals of their same species rather than with somebody of another species. And this is because they are competing for the same niche with individuals of the same species. So in general, people have this feature that this epsilon is larger than 0. And another thing you can add is some immigration. If you think about a place in the ocean, you will certainly have that some species are brought in, but you can think of it like exit from dormancy or speciation. So this is a feature that depending on the model you will have a different form of immigration. What this immigration does is that basically it prevents species to go permanently extinct, which would, little by little reduce diversity. So I want you to pay attention that this noise term actually has an important parameter here that is somehow setting the time scale of competitive exclusion. So this is the product of, so tau is the correlation time of the noise, and sigma is the variance of the fluctuation. So the larger is this time scale, the longer it will take, the shorter it will take for one species to take over, or if it is in competition with another one. Now let's see what happens in the simplest possible case. All species are statistically equivalent. Even though at every particular moment the growth rate will be different from one species to another, let's choose these growth rates so that the average over time of the growth rate of every species is the same. Okay, this is so like a neutral hypothesis. The second neutral hypothesis is that all species get kept from above at the same rate. So they respond in the same way to the total abundance of the community. So if you simulate this system, as you see, these are the growth rates. So they oscillate around the same value, and just species will be different from one another. So to what extent are they actually equivalent or not? And so recently, people have proposed that actually this is enough to maintain a lot of diversity. And indeed, what you see is that after a while, the system seems to engage in a sort of turnover dynamics where you have one species that grows and the other one, and then it feels like actually if you look at the abundance distribution, you will see that this is actually broadens progressively. So you start with something that is very peaked, everybody has the same abundance, and then they spread out. The problem is that if you keep simulating the system for longer times, actually this turnover dynamics will slow down progressively. And at a certain point, you will get that only one species is dominating, and this one may switch to another species, a dominant species, but it is only one that is really dominating the system. So basically, your whole diversity has collapsed to one. So the other problem is that this exclusion time scale is actually rather fast, so you can't really overcome it easily by increasing the number of species you add. Even if you add many species, you will collapse to this one species state pretty quickly. And the explanation of this can be, in this particular case, understood by an exact mapping of these equations to the random energy model that has been studied extensively in statistical physics, where you can understand this transition from a very diverse state to a mono-dominant state as basically a phase transition of condensation. That happens because the formal solution of the equations is actually in the shape of a Boltzmann distribution, where the exponent here is the integrated fitness over time. So despite the fact that all species have the same distribution of fitness, so the fitness undergoes a random walk, the integrated fitness is actually different between one species and another one, and in the end, the species that has the highest time integrated fitness is the one that will prevail. So how do we get out of this? So this cannot be the only solution. I forgot to say, so this is what people call the stickiness of rarity. So this first part, the idea is that the more you are rare, the slower your population grows, and therefore you remain longer close to zero, but then sooner or later you will grow again. Yes?
Okay, but why is this a problem? Meaning, maybe in reality, the time scale is so long that we haven't reached that yet. Right? right?
It is possible,
the
yeah.
assumption here is that we are well beyond the typical time scale, but that doesn't need to be the case, right?
Yes, we don't know. Let's say that by itself is not guaranteeing you to maintain diversity. Whereas this was the point of that publication, was to say, okay, we don't need anything else. It's not true. So it's a transient state. And then the problem is, okay, is there a way to stabilize this transient state? And actually, there is an easy way to stabilize this transient state. And this is corresponds to other hypotheses that are commonly done in ecology. So this would be the distribution in the case where the completely neutral case, but even if you keep neutrality in terms of the average fitness, it's enough to introduce some immigration and some self-limitation to actually stabilize this transient. So the equations now look like this, like those I was presenting before. So I keep, for the moment, that the average growth rate is the same, but I include either self-limitation or immigration or both. And the duration of the transient, if you if you introduce just one of the two, the duration of the transients become much, much higher. So you go up orders of magnitude, and then your argument is perfectly valuable. You know, perhaps if the time scale of competitive exclusion is the age of the universe, then is not really relevant. But the interesting thing is that if you include both of them, actually you go into a regime where this transient becomes the steady state. So you never actually go to monodominance. And what you obtain is something like this, where you have forever this substitution of, species that become dominant for a while, then they seem to disappear, then they come back again. And this is actually something that looks a bit like what people have observed when looking at communities at a very fine taxonomic resolution, so typically at the level of OTUs. So here you have some observations over several days, but as you can see, there are some spikes in the presence of certain OTUs. This is a coastal station, and this time scale is shorter than the seasonal time scale. So this was observed also with morphological studies, that actually when you look at the species level, you find much more variability than when you look, for instance, at the level of a functional group. And if you look at all the time series that have been published in ecology, some people have started to analyze this with non-parametric methods. And it turns out that the phytoplankton time series seem to be very often prevalently classified as chaotic, so to have this kind of intermittent behavior. So I could talk a long time about this. If you're interested, we can talk about it later. But I would like to go back to the simple model and just focus on what can you obtain if you have all these time series. So suppose you have the time series of many, many different species and many, many different times. Of course, we don't have this in empirical data, but we can do this with simulations. So we can look at two different things. We can either have like you are sampling in the water at a given moment, and then you have the abundance of the different species over time, and this histogram that is the species abundance distribution. Or you could also take one single species and then look at what is the distribution of the fluctuations over time. Okay, and this you can do in principle for every species. You will have a different distribution, but it turns out that if all species are on average equivalent, you get the same distribution. And this distribution that is called the fluctuation abundance distribution happens to coincide with the snapshot distribution. So I will mostly use the two things interchangeably for now. And let's look at what is the shape of these distributions. I forgot, sorry, to put this, to group this with a figure. Anyway, forget the letters. So now, if you go back to the equations, one Emil realized that actually you can reparameterize the equations. And if you do this, instead of having 7 independent parameters, you end up having just 3 compound parameters that are important for the dynamics. And these compound parameters are related, this one on the ratio between the exclusion time scale and the average fitness, growth rate of the population. This one is related to the self-limitation, and this one is related to immigration. So whatever are the parameters you choose for your system, what will matter are just these 3 parameters. And this is to illustrate this. So you fix these 3 parameters, so you provide some values, and then you randomly pick the real system parameters so that the compound value is the same or is very similar. And what you see is that actually the shape of the distributions also is very similar. Actually, it's even more than this. This has been obtained by fixing the ratio, the log ratio of these parameters, so only 2 compound parameters. So somehow you have many different models and many different processes that end up giving you exactly the same distribution. This is the first observation. The second observation is that actually these distributions they don't have always the same shape. Depending on these 2 compound parameters, they can go from something that looks like a power law to something that looks normal, something that looks like a gamma. So these are all the kind of distributions that have been used in the literature to fit the data. How can one understand this? So there is the nice thing about this. Oh, sorry, I forgot to say these famous letters. So the 2, let's say, 2 extreme cases are this one where you have that the time scale of competitive exclusion is shorter. And this one, so that is called, I mean, we call it EE because it's like exclusion. And this is where you have a sort of a balance between self-limitation and buffering. I will explain this a bit better later, but I wanted to introduce the letters. So how do you intuit why the shape of the distribution is changing with just these parameters? So if you think about this system, so this is the abundance. These equations, the equations I was presenting, they describe a sort of a random walk that happens in this space here. So you can hardly go below the immigration threshold because then you will be put back by the immigration. You cannot go above the carrying capacity of the whole community, but also as a single species, you tend not to go above a value that is below the carrying capacity that is due to the additional self-limitation. And the distance between these 2 lines basically goes like k times epsilon. So somehow you have this process that is drifting inside here. And what can happen to such a process? There are not 10,000 things that can happen. So if your growth rate on average is negative, let's say your effective growth rate, not the R star, let's say you have a trajectory and then you compute the realized average growth rate. If this is negative, you will have that the density accumulates close to here, and then you will have some kind of power law decay. If instead your average growth rate is positive, then you will grow, and then when will you stop? You will stop either because of self-interaction or because of interaction with the rest of the community. So this being said, it is possible to find a much simpler equation, that is the equation of just one variable, that produces basically all the shapes that you see. So you don't need to have S different equations. This is called the stochastic logistic equation, has been proposed as a heuristic model by Jacopo Grilli to explain his gamma distributions. And we have proposed it also as not as a heuristic, but as an approximate solution in the case of power decays. And the important parameter here is this effective growth rate, that is this long-time average of the growth rate, but that you cannot connect very simply or directly to the parameters of the original model. And similarly, so here you will have, an effective variance of the fitness or of the growth rate, and also an effective correlation time of your fluctuations. And this applies, in this case, it was applied to a deterministic model. So you can compute this also in deterministic models. The good thing about this is that it can be solved, at least in some approximations. And from these approximations, you end up having this distribution that is basically the one I was fitting before, where explicitly you get the slope of the power law distribution. That is a function of these effective parameters. And as you can see, so this slope this should be a plus, sorry. So this slope, it appears to be vastly between 1 and 2. That is exactly the range that people have found in this big fitting exercise with all the data we have. So now, if we want to characterize the shape of this distribution, we can do it a bit more quantitatively by looking at the width, the support of the distribution, the model value, and the slope. And this is just to show you that what I was showing as an example is actually true in general, that this two axis, so this buffering axis is the log ratio between two of the parameters I showed before, and the stabilization axis is the log ratio of other two parameters. It is actually organizing in a gradient the shape of the distributions. And these are the 4 points where I showed the specific distributions. So you see every point is one simulation with randomly chosen parameters, and all that matters in the end are these two axes. So this part here is gray because this is the power law exponent that is computed from the approximated, so the single species model. But this single species model is not a good fit in the region where you basically don't have a power law, and then you can't really fit it decently. So just going back quickly to the empirical observations, so you can get with this model everything between a pure power law to a gamma or a log normal. So there's no point in really discussing what is the best model, because what differentiates a model that has this and this is not actually one single fine detail, but it is a combination of parameters that can be changed independently. So if you change 4 parameters independently and you keep these ratios the same, you will get the same distribution or not, depending how you change them. So all this, all I showed you is for the case where all species have the same average growth rate. This is not the case for sure in reality. So what we try to do is now let's take growth rates, average growth rates that are distributed. So they, they can be pretty spread out. And then we looked at the abundance fluctuation distribution for single species independently. So here you can see that, okay, you transform these simple distributions in something that is actually the overlap of different distributions. And this overlap is such that the species that are most abundant tend to have distributions that look more like a gamma or I don't know. And something that people usually don't say is that in this works, they use the data from the repository, I forgot the name of it. Anyway, in this global repository, they get abundances of microbes, but they don't want to put in microbes that perhaps don't really exist. But they only take those that are actually with a high occupancy, those that are in general abundant. If there is some observation of a OTU that is unknown, they don't put it into there because they are not sure whether this is really an artifact or a species. So somehow, if your data set only contains the species that are up there, then you will observe rather gamma distributions. If you take the data like those we took, where you have all the OTUs, including those where people have no idea what they are, then you get this. So another observation that was proposed long ago, that was put forward long ago by in this study on fish, is that actually, if you take an abundance distribution, this might be the overlap of 2 abundance distributions. And these 2 abundance distributions actually differentiate the species that are locally present, so endemic, with respect to species that are visitors or occasional. So in the case of fish, they knew what were the species that were endemic and what were the occasional species. And indeed, they could find that if they were separating species that were present every year from species that were present every now and then, they would find the 2 different distributions, and the species abundance distribution would be the overlap of these 2. Unfortunately, we can't say the same for plankton. We can't know what is endemic and what is not, but it would be interesting to know. And finally, the last point I would like to make is that with this very simple model is also explaining why you have this kind of decay in how similar communities are in the course of time. So here you start from a community, and then you compare this community with all the other communities that you sample over time for a long time. And what typically happens is that similitude. Similarity decays over time, but it doesn't seem to go to 0. And in the model, this is also what happens, but it depends on in which regime you are. So if you are in the regime where you have the power law distribution that is associated to a lot of turnover, actually this similarity goes very quickly towards 0. Of course, this is not true anymore if species start to be different from one another. So here, the pink line is when you have perfectly identical species, it would go to 0 in the end. and when you have that some species are on average more abundant than others. But this is much, much more pronounced when you are in a regime where actually species tend to be on average viable and not on their path to extinction somehow. Okay, so time to conclude. So this was a very simple model, but I think really useful to understand what is the effect of the combination of very simple ingredients on the distribution of abundances. So here we have considered basically three things: fitness fluctuations, immigration, and self-limitation, which are, in one way or another, the three basic ingredients of many, many different models. So what are we left with? If we have these broad classes of distributions that are, you can go continuously from one to the other. So the fact that you can fit one of these distributions is not really telling you much about the system. But actually, if you have already time series, then you can hope to get some more insight, and possibly the combination of different kind of data can help us, if not really knowing what are the exact processes that underpin the distribution, at least to identify what class of ecosystems a given community belongs to, and for instance, whether some change in an external parameter can drive you from one kind of ecosystem to another. And this is here. Thanks, this is all. Thanks a lot.
PhD first.
Absolutely.
I think my question is a bit tautologic, maybe, but like in the limit of a very big ecosystem, like you would consider the gut, but if you start to make your ecosystem bigger, at a certain point, lambda will be zero, because at a certain point, maybe you talk about the entire Earth, you cannot have, so
then you have evolution.
Okay, then you have speciation. So this is a problem that people, like, that
I mean, so some people, they just forget about this existence of this problem, and they just put all the data they have together. So for instance, in the study of tar oceans, so if you study a local sample, you are really looking at what is there. But some people say, okay, we have these abundances that are, by the way, relative abundances and not absolute abundances. Let's put together the abundances of everything we have observed in the tar oceans. And then you put together things that are very far apart, and then you would have, in principle, the meta population or beta community level. So if you do this, indeed, you find something that is unimodal, that is a prediction for meta communities where you can observe either at the local level or at the level of the meta community. The point is that this exercise, strictly speaking, doesn't make sense because you cannot aggregate relative abundances with one another unless you know what is the absolute abundance, which is not something that you know in this kind of data. So this is why I always insist on the fact that one has to look at, sample by sample, unless now people are starting to introduce like common standards, spiking methods to get actually the absolute abundance of these samples. But this is not, I mean, it was certainly not done for this kind of data. So yes, the expectation is that when you look at the level of the whole Earth, you should get some unimodal distribution, provided that your speciation is not fast enough. If your speciation is fast enough, then you would also get power law.
Yeah, that was the same question that I had. But I also had another question, so I'll get my second chance. Which is something you alluded to at the end. So I was wondering whether in one regime or another regime, let's say the susceptibility of the system to external perturbations is significantly different, so that if you are in one set of parameter space, maybe you're more susceptible to then move because things change.
Yeah, I mean, I would imagine so. I mean, I haven't looked at this, but I would imagine so, because if you are in the regime where your effective growth rate is positive, you are fluctuating around the positive value, so you can get kicked and you will come back there. If you are, on average, you are doomed to extinction, and you are kept alive just because there is some process that brings in new diversity. There, if you actually get rid of 50% of the species, then the system would collapse. So it depends. I think it depends how extreme you are in this space. But it's a very interesting question. I think we should definitely look at this more detail.
Yeah, I just wonder how size fits in all of this. I would assume that it fits through growth rates, which are probably related with size, but traditionally there were like, those size distribution in the ocean, etc.
Yeah.
So,
I'm not sure how. No, I don't know, actually. I'm used to think about the size as something that could determine, actually, in some other models that actually reduce to this one in some limit case. So models like the Lotka-Volterra equations, where you actually model explicitly the interaction between species, then size distributions would result in a power law distributed traits. And if your interactions depend on these traits, then you would see this in the abundance distribution. But exactly how to introduce them here, I'm not sure, because so with size you would have fragmentation and aggregation processes that go on, but you don't have this multiplicative noise that you have here. So all these effects come from the fact that the growth rate is defined at the level of one individual. So you have like per capita growth rates. But yeah, I'm not sure.
Yeah. As it's just an idea.
Yeah.
I would assume it was smaller species are more abundant generally, and they have higher growth rates.
Yeah.
So I would see that there's some sort of.
You mean that they? Yes. Yes. Okay. You're right. I didn't think about this. So species that have faster dynamics, then there are those who should be more towards the rarest end. Is that true in reality? So in the Tara data set, the smallest, we had 4 size fractions. So the smallest size fraction was the one that decreased the slowest. Was it? And larger size fraction, the decrease was. Okay, I should look into it. Yes, it's an interesting thing to look at. Thanks for the idea.
I have a question which I don't know if it's a question, but you said that the phytoplankton species succession essentially is it was the most chaotic between the ones that you the other organisms that you consider, and I think this is a problem that in ecology, like many marine ecologists ask themselves, of this problem of successions.
Yeah.
And I think that usually the answer that when people measure, etc., is that it's an extremely multifactorial process in which you have like 1,000 of parameters, temperature, salinity, cell toxicity, that makes like one species win at a certain moment. So I was wondering if it's there's a way to do from the data if it's truly chaotic, that a species wins because of a random fluctuation, or if it's simply like a problem of not knowing.
No, it's a very, very important point. So the data, the analysis of the time series is not me who did it. It's a group of Steve Munk and especially Tanya Rogers who did this. We are collaborating with them actually to try and see how their algorithm performs on some numerical simulations and see whether they can actually detect correctly the existence of chaos or not, because we can decide what things are chaotic and what are just fluctuations. But generally speaking, so again, there are many different models that end up giving you this kind of intermittent behavior that sometimes has this succession structure. Some are stochastic, some are deterministic. In the end, even deterministic models, if you have many species, they behave like stochastic models. And so you can't really tell from the data which is which.
Yeah, maybe it's more philosophical, the question.
No, but it's
it's
a super important question, and there are people who are looking into it and trying to figure out, can you tell if actually some fluctuations, for instance, they come from fluctuations in the biotic or in the abiotic environment? I mean, we all know that in the ocean, the abiotic environment has intermittent fluctuations. But then, is that enough to describe the fluctuations that are really over the timescale of days for these communities? Or do you need, on top of this, to include also interactions with the rest of the community? So, since fluctuations are seen also in communities in the lab, I tend to think that actually it's a combination of both, so that you can also have fluctuations that are driven purely by the interaction among different species. But this is, there are difficult experiments to do, so people don't agree on this.
Yeah, I have a couple of questions. One is about these fluctuations. I'm wondering if you've tried playing with different patterns of fluctuations, because different sizes will experience environment in completely different ways. And then the other question is somewhat related, that all these very rare species, at least thinking about, for example, bacteria in the ocean, they will probably be dormant or very almost inactive, no? And how do you incorporate these in the model? Is it because I mean, at the end of the day, we think that it's not completely neutral, no? That they are kind of waiting for their moment.
So there are models for dormancy. We are also studying one with the lot-Cabothard equations, multi-species lot-Cabothard equations, where species are allowed to go dormant. And then again, depending on the hypothesis you make on dormancy can actually increase or decrease biodiversity, which was a bit surprising, because people usually thought, okay, if you add the dormant stage, then you will increase biodiversity. This is not automatic, because actually you need some specific conditions for dormancy to have a positive effect on the overall biodiversity. Sorry, the first question was about fluctuations. Yes. That's definitely something that we should look into on adding an external forcing. So what we've done so far is that this noise that we add, so you can change, for instance, the correlation time scale, which is just the first, because usually people add like white noise, which is just one kind of noise and not very realistic. So the time scale of the fluctuation is not particularly important by itself. So what is important is this combination of parameters. So how fast, so what is the correlation time scale, and what is the variance of the fluctuations that combine in this. Yeah.
In fact, you could actually go back and infer from the time scale of the variations different parameters like I don't know, growth rates or number or abundance of the species. And you go the way backwards.
Well, I guess that if you had a time series that is very resolved, you could have the distribution of growth rates, for instance, for different species. That I think it would be feasible in principle, but measuring growth rates in the wild is not easy. And no, definitely, I think it would be very important to add like seasonal fluctuations because some of these patterns synchronize to these external fluctuations, and I bet that would be different for a purely stochastic and deterministic system.
Do you think that by looking at a particular shape of a distribution, you could say something about the physics, the regime of fluctuations that they are experiencing?
You can probably say something. The problem is that we should actually do more comparison of these shapes across ecosystems. And this is, so people who compare across ecosystems, they usually want to find a universal pattern. And then, they want to see what is the same everywhere, not what is different from one place to the other.
Yeah, I'm asking what is different.
Well, what is different, for instance, there have been people who claim that this slope of the power distribution depends on features like, ideal kinetic energy. So they analyzed exactly our same data set, and then they were saying, okay, now, this is variable. So the point is that they were fitting this data with just one power law, including all the data, so the rare part and also the common part. So they got much more variability than what we get. And then, yes, they found some kind of correlation, but when we looked at our data for the slope of the rare species, there was no significant correlation that was consistent across the data set. Because if something doesn't vary, then also it's very hard to find this kind of correlations. So I think that there is certainly, and it is to be expected, a correlation between the oceanographic conditions and the species that are blooming, blooming or that are highly abundant. Because these are really species that are sort of, selected for in that specific place. These are sort of the endemic species. But then in the ocean, this endemic species, sort of patch, has a finite lifetime. And then after this lifetime, the species get dispersed, they end up somewhere, they won't disappear because they are so numerous that in the end you will find them basically everywhere in this background of their species. And then so what shapes this background is not really what happens locally. It integrates a phenomena that happens on a scale that is much larger.
What about if species need sexual reproduction to succeed? As opposed
I don't care about sexual reproduction. No, it's just too difficult for me. Yeah, it's something I know nothing.
No, because when when you, unfortunately, to reproduce sexually, you need to encounter someone, no? Because, but when you don't, then you can bloom anywhere, anytime, no? So I guess,
Yeah.
yeah,
I don't know whether sexually reproducing species have. I mean, I think that many species that reproduce sexually, they also have asexual ways of reproducing. In microbes, in many cases, you don't only reproduce sexually, otherwise you're dead, I guess. But I mean, that's pure speculation. I really don't know. I think that for many species, also people don't know how exactly they reproduce. I mean, they know for certain species that have been observed.
Thank you.
Thanks a lot.